So it's a great pleasure. My name is Zhen, and my current role is the leading oncology at EMD Serono. But today I have an honor to serve as a physician scientist to moderate this panel and work with my prestigious colleagues to take you through this topic David just mentioned, specifically what rare disease registry can help with medicine development. So before I start, I do want to, on behalf of my panelists, to take the opportunity to thank the organizers, Bill, Andy, Julia, to host this meeting during this challenging time and do this virtually. Many of us are frequent flyers here, been with the PDF FDA meeting over the past 9 meetings. I think most of us have been participating in each one of them because we love this meeting so much, giving the importance, the topic, the cutting edge topic, and most important thing, I think, like David said at the beginning during his keynote speech, it is unity. Because unless we have this open dialogue, this unity, we won't be able to advance medicine. Using my prestigious colleague, Mace Rosenberg always says, there's a speed of oncology because we want to develop a medicine for patients. So big thank you for that. Also, I do want to take a moment because this time we have this virtually. Many of the people are sitting in the audience, sitting in the audience as virtual participants. I do see Dr. Murphy there, and also the founding person, my colleague Mace Rosenberg. I do want to take the moment to thank them for starting this tradition and with Dr. Pasteur to enabling this very collaborative effort that really help us advance care for patients. So this specific topic of rare disease, very near dear to my heart as an oncologist by training, is I think that when we notice large indication, when we notice that the medicine development had to take a significant term in terms of acceleration, there is a population, there is a patient population that very difficult to reach and very difficult to conduct clinical trial with, and that's the rare cancer patients. And today's world, with the advancement of genetic, I would say capability, measuring the next generation sequencing, also there is a population called genetic or rare genetic alteration population. I still remember I used to read a book of N1 clinical trials. I mean, we're going to enter into an era that every patient's a rare cancer patient because the rarity of their background. So we have to treat each patient's care with differences. And that's something I thought really, really important. Right now, we had this inflection point from both angles. One is, I think over the past 3 or 4 years, I'm going to pass on to the panelists. In terms of the progress we have made, I think it's the moment to celebrate a little bit of this success and the progress we made as a field. We see medicine approved based on the rare disease registry, real world experience, and also using that disease registry serve as a natural history, to Greg's point, to help the regulatory decision making. But also we wanted to look into forward to, I think my panelists, Michael Wong, challenge us, say, what about action? I think I really like this notion that also start by David, say, hope need to trigger action. I think this is something really important we will touch on at the end of our talk, and really also lead into the way of my great friend, Sean Cozen and Josh, to talk about a example of the fireside chat. So with that said, maybe we'll get started talking about speed oncology. What I'm going to do a little bit different from David Reed when we started is I'm going to let the panelists introduce themselves and then quickly touch on actually your recent success and experiences with the rare disease registry. There's no better person to start this with. Mike, I'm going to pass on the stage to you. Thank you.
Thank you very much for a kind introduction, and it's an honor and a pleasure and also a privilege to be on a panel with such esteemed colleagues and to be able to speak to you in general. I'm a professor at MD Anderson in the medical oncology department. I specialize in skin cancers and Merkel cell carcinoma in particular. I also serve a role as a medical executive within the MD Anderson Cancer Network, which comprises affiliate and partner hospitals throughout this country and the world. And in that capacity, I'm responsible for integration and special programs. And so I have a footing both in sort of a local thing where I touch patients. I had clinic just yesterday, and also more globally. I'm just going to say one word about this. And, I came to this in a way, serendipitously. I was at a meeting that Dr. Brownlee from the NIH arranged for a workshop in Merkel cell carcinoma now almost 3.5 years ago. I stood in front of my colleagues and said that one of the major missing points is sort of understanding of this rare disease, and that a registry and a database of sorts would be essential, and that I would not come back to the meeting if I didn't have one set up. So I'm very fortunate to be able to work with Dr. Miller, which you met in the previous panel, to really put this together. I'll speak more about this later in detail, but I'll pass the baton on to my colleagues for their introduction and remarks.
Greg, maybe we'll pass on that stage to you.
Again, it's a pleasure to be here. My name is Greg Friberg. I am the lead of the development organization for oncology and bone at Amgen currently. I'm a medical oncologist by background. Had had a short academic career before leaving for my industry role about 14 years ago. I've had the real pleasure in more recent days of working on some projects that leverage real-world evidence registry data, particularly in the ALL and the MRD, minimal residual disease positive subset, very rare cancers. And again, I think we're gonna get into these themes, but this concept of is that data there to supplement and complement, as Dr. Bieber, I think, pointed out very nicely, or can it be used for something else? And of course, the devil's in the details, and looking forward to having additional conversations in the panel.
Thank you, Greg.
Hi, good afternoon, everyone, and thank you so much for having me and my other FDA colleagues here to be part of this very important discussion. I'm a pediatric oncologist and the acting deputy director of the Division of Oncology 2 in the Office of Oncologic Diseases at the FDA. And within our division, we have the team that oversees the regulation of cancer drugs and biologics that are intended to treat pediatric patients with solid tumors, as well as patients with brain cancer and other rare solid tumor cancers. So this topic of rare disease registries, cancer registries, is really a very important one and near and dear to my heart. With respect to use of registries, I think others have talked about this already, particularly Julia and Gregory. And we have had a very long history of using registries for use of trying to characterize safety, particularly in the post-market setting. Particularly when we're concerned about rare adverse reactions that may not have been adequately represented or even seen maybe in an initial database, which, it certainly can happen when you're looking at small data sets for patients with rare cancer. And so we've been using registries for that purpose for quite some time. And additionally, as already been mentioned, we have used registries many times to help inform the advice that we're giving to sponsors when they're developing drug development programs and clinical trials to help provide the data that will ultimately support a new drug application, to help us understand endpoints, provide advice on target effects, et cetera, in that context. And although these aren't examples of use of data from registries per se, we have had precedent for use of real-world evidence derived from data sources outside of clinical trials to provide additional evidence to support the approval of drugs to treat cancer. Notably, a couple just quick examples were in 2014, the approval of blinatumomab for the treatment of Philadelphia chromosome negative relapse refractory pre-B-cell lymphoblastic leukemia. We used data in that approval not only from a single-arm clinical trial looking at complete response rate, but we also looked at data extracted from 2,000 patient records derived from data from the European Union and U.S. clinical study and treatment sites that gave us patient-level data, which provided very important clinical context for us to assess the meaningfulness of the CR rate observed in the clinical trial. So we use real-world evidence there in that context, and additionally, more recently, in the 2019 approval of Ibrance or palbociclib for the treatment of male patients with hormone receptor positive, HER2 negative, metastatic or advanced breast cancer. We use data from the Flatiron database to help document real-world response rates, and a small number of men with breast cancer, I think it was 20-some plus. But using that data, in addition to FAERS data to help provide some safety information, claims data were used, all within the context of what we already knew about palbociclib from randomized clinical trials in women, put together a data, and enabled the construction of a data package that ultimately led for, approval in this very rare, but important population of patients with breast cancer. So, I think all of those principles really extend to and speak to the importance and utility of registries, which I think provide even more potential in many ways to provide some structured data that we might need in the future, that we will need in the future.
That's really great. Yeah, and thank you, Marcia, for a nice, summary and also list of recent successes and how, from the agency was able to tap into this, a rare disease registry or real-world data to enabling, I think, medicine development for some of the most rare cancer type we're seeing. Mike, you want to make a comment?
Yes, yeah. I think just to put a few things in perspective and a slingshot of what Dr. Reese said in a previous panel, and both Dr. Donoghue and Dr. Friberg spoke of specific patient population. In one, she mentions, failure of a chromosome. She mentioned a hormone receptor positivity or negativity. Dr. Friberg mentioned minimal residual disease. Where I'm going with this is that we are in a world where it's going to get rarer and rarer, where we're going to personalize it down to the molecules. And if I learn one thing from Dr. Frankenbaum, there may be a Castleman disease, which is IL-6 negative, mTOR positive, VEGF receptor positive, ligand negative. And that's how we're going to start defining things. And if so, what's going to happen is that this is not just going to get better. This is going to actually get worse as we try to fractionate these drugs. And why do I even mention that? Because nowadays we sort of use medicines without biomarkers in the hope that it's going to work. And I built my career in immunotherapy, and we give immunotherapy to almost everyone who fits the clinical criteria. But we know that only less than half of them will have a long-term response. And if we only had a biomarker, I want to say not biomarker, but biomarkers with an S. So as these things happen, we're going to need some mechanism to really flesh out who's what. I mean, this is sort of looking into the future, but I don't think it's going to be the distant future. So more than anything else, I think registries will become increasingly important, both post-marketing and in the initial drug development.
Love it, because that's the thing that we were excited because this is going to become a mainstream. I think it's no longer this unique one-off situation. We had to deal with say, open mind and also some of the exceptions, but now it's going to become a mainstream. Greg, you want to make some comments? I think your recent experiences could also give a flavor to what the lessons learned and also some of the successes.
Sure. Thanks, Zhen. Particularly with a disease like ALL, and particularly if you look at adults, it's a rare disease. Registries really have opened the opportunity to understand the disease in different ways. Of course, I kind of think of it in three bins. Any registry data, it'll have its challenges, and I think we'll get to that in answering a separate question. But they can be used to reflect on how the patients are doing in the past. You can think about which patients are being well served and which ones are absolutely not being well served by the therapies that are out there. And in the example I cite, being MRD positive, for example, is a very strong predictor. People who weren't okay, you needed to think about doing something. Similarly, when we're at the end of, say, we have experimental evidence in hand, you can think of this registry data, this external evidence, as being used to put that into context. I think we've discussed that already. There's a really, I think, clever way to use this data that's probably evolving, which is could you think about a way to use it as a prior evidence base to inform the actual what goes on in the middle, which is the testing of new therapies, to be prior information and in, say, a Bayesian approach, use it to either inform your control arm on a very basic level. We can use this data to, again, pick entry criteria. Think about who should come onto a randomized study if you can do it, and who should or should not be excluded. But really this idea of using the data in a more adaptive way is something that I know that several groups are thinking about. And again, it goes to show you, depending on what that data set is, where it comes from, how much confidence you have in it, you could think about using it in different ways. And Not to say that anyone is trying to make randomized studies go away. Probably the greatest invention in the history of medicine is the randomized control trial. But there are just some settings where it will be impractical, if not impossible, because of numbers, where patients are, or patients aren't living long enough. Very sad practicality that you won't be able to get them to that therapy. So again, some thoughts off the top of my head, but I think ALL presented a nice case example where the first and the second, thinking about afterwards and beforehand, putting into context how you're going to design a study and then what it means were used. But the future will be using that data in again in the study and incorporating it into the statistical design.
Thank you, Greg. I'm going to write it down. The randomized control trial is still gold standard. maybe pass on that to Martha. Maybe let you give an agency's flavor in terms of how you see the world evolving. Martha, maybe I pass on to you and see how you view agency view where we are leveraging the real world data, either mainstream or how it can complement the mainstream.
Thanks. I was half joking when I did this. And I appreciate, we do, of course, believe that the gold standard is a randomized control trial because it can control for sources of bias that we may not be aware of, and particularly it's really good at, when the sample sizes are big enough, it's really good at kind of, mitigating bias for factors we know about, and very importantly, ones that we may not know about that could impact patient prognosis. So, I think there are, many circumstances, many where they're still very relevant and feasible to conduct. But, we do recognize at FDA that there are situations where randomized controlled trials are not possible. So either because the patient population is so small or because there's no good comparator arm and it wouldn't be ethical to randomize patients, for instance. And so, we've relied, in oncology, a lot on single arm trials, looking at sort of concrete evidence of tumor effectiveness stemming from response rate and durability of response, in particular when we're looking at rare cancer drug development. And so, the way I view use of registry data and other sources of real world evidence at this point in time is it's hard to make a general statement about it because it's a little bit really important. The context of use is important, and the particular situation, the particular disease is really important. And, and this is my own personal opinion, I don't yet see reliance solely on the use of registries at this point in time, as something that is likely to support a drug approval. But it's very easy to see, and we are already having multiple conversations and are overseeing drug development programs where real-world evidence from registry data and other sources is really providing a key and very important component to the data package that's contributing to what will hopefully be demonstration of the drug's effectiveness for rare cancers. So, I think in a particular situation where registry data and other real-world evidence data can be important is helping us interpret when there are circumstances where we can't rely on response rate, or we may need help interpreting what response rate really means to a patient. Having additional data that describes patient outcomes, both in patients who've received the drug or patients who have not received the drug, to help provide that context to us when we're interpreting the data that we have is crucial, and I think will be continuing to be leveraged more and more. There's been a lot of discussion and a lot of work thinking about using real-world evidence, as an external control, a synthetic control to help us interpret time to event endpoints in clinical trials. And I think there's a potential avenue, certainly for use of registry data for that purpose, although, it would have to be very, very, very carefully thought about and constructed, and, would require a lot of interaction and discussion on our part to make that, a critical component of a drug development program. So, I do see that there's going to be, as Greg, I think, mentioned in the last panel, I do think that there's going to be more and more focus on use of these types of data in the future. And I think particularly, now that we're looking at rarer and rarer cancers and rare subsets of rare cancers, which is driving, changes in our drug development paradigm. And I think thinking about use of registries in a global sense to capture data from as many patients as possible to give us, confidence in the results will certainly go a long way in helping that utility too.
That's really nicely said, Martha. You actually helped me in my job to open the next topic in terms of discussion about the challenges. So, and I know that Mike's very passionate about, sometimes, when you look at, to your point, Martha, the disease-wise is so rare and make something impossible to do, find that many patients to do the randomized trial. And also sometimes, from timing, the cost and everything, the efforts, they would not be, enabling a sense of urgency to accelerate. Because we know the cancer patient can't wait, and cancer is a lethal disease. So, and how to balance those two when the science advances very quickly, and we have a certain step to reach to a regulatory science to enabling understand the risk benefit, and this has become an art, I would say. And then I look at Mike. I mean, probably you can give us your perspective of the challenges, particularly moving forward, because. And also the dialogue between the academic and the agency. So regarding how to overcome the challenge to a certain degree.
Sure. I when I was a much younger investigator and I had hair and a ponytail at the time, I see everybody laughs when I say that, including my kids. But at that time, we used to see, the FDA as an adversarial sort of agency, right? If only to let us do this, why can't I do that? But the reality is I've come to realize that this, it serves a very important role in sort of making sure the answer to two questions to me. Is it safe and is it true, right? And simply put, that's what it is. And we have to rise to that bar. And so I understand that. I also want to pick up a point that Greg built up, which is, these registries are not just sort of hypothesis testing, but they can be hypothesis generating in a sense, right? And our keynote speakers, sort of mentioned that in spades. And so, is there something down at the local CVS that we can see? So why do I say that? Because there are drugs that we know that overall survival in many of our patients is a function not just of what the test drug they got, but of the downstream care that they got subsequent to that. And as that improves, that can differentiate one arm over the other. So capturing that longitudinally, as Dr. Miller said in the previous panel, super important because now you start getting clues. And I, for one, believe that if you truly understand immunotherapy, sequencing matters. Where you go with your, is it a vaccine first? Is it going to be a checkpoint? Should it be a cytokine? Now we sort of give everything because that's all we got. But as we get more sophisticated, these things become increasingly important. And so the challenge is I have to be honest with you. I came to this because of the fact that I think it would be impossible to mount a reasonable phase 3 randomized control study in something like Merck, which I treat, because of rare disease and also difficult getting patients and also the treatment benefit. Or you have a drug that's 10 times better, then of course it's a small trial. But I think the blockbusters and paradigm-changing drugs only come once in a while for ample reason. And because of that, everything else will be incremental. And you can imagine the number of patients necessary to make it happen. So know that will be really difficult to do. And lastly, I think there's a voice here that's not heard, but I think David Fangenbaum brought it out, which is the voice of the patient, right? They demand that we do something now. So how do we do this in the context of what we have in rare diseases? So the challenge is ours, right? And so one of the things Dr. Miller and I have been trying to put together, and particularly David, because I'm sitting on his shoulders and hope he's not cringing as I paraphrase him, is to really build sort of robust databases in collaboration with stakeholders like pharma, like the FDA. And really it's like a limbo dance, right? How low can we go in the sort of quantity and perhaps quality of data that we get to satisfy regulatory requirements? And again, this is not adversarial, but we understand where you're coming from. How low can we go in the number of patients that we need, right? And are we able to build an adequate synthetic control arm? These are all challenges for us, right? I'll stop there. These are sort of mainline things. There's of course, I have a basket of these things, but for me, these are the top line items, right? We have to solve the problem today, right? I had clinic yesterday, and we have patients coming in, and what trial they have for me now. So for me, I feel this pressure every single week as I see patients over and over again, and I'm just passing my anxiety to you. I don't have the answer, but here it is.
My job is simple. I'll pass that right to Greg. he's one I always can count on. Like how to overcome this, given that you have recently experienced this and brought in some success and also treatment options for ALL patients. Greg, how you look at these challenges, safety, efficacy, and also the quality and, inherent bias with the disease registry.
Let me pick up on that bias comment that you made. We often talk about how are we going to eliminate bias, but I like to use the sports analogy. You can never eliminate bias. You can only hope to contain it and understand it. And from that standpoint, really, when we're looking at these sources of data, of course, where you're getting the data from is absolutely essential. And we think about, well, what information is important? And I think, again, our keynote speaker very nicely laid out, loved his slide that had the 5 bins of the different types of registry. I stole it, and I'm going to use it in the future. It's just a very nice, I think, illustration that the choices you make will have consequences, whether it's on cost, on quality, or on numbers. But there are a few factors that I think about when you're looking at these registries that are probably universal. And one is that you have to make sure that you have the fields, that's the data information, the structured data, the fields that matter. And of course, what matters to you will depend upon what questions you're asking. But at the very least, since we're almost always asking time-based, how did these patients do over time? You need the basic prognostic factors for studies. Now, with ALL, for example, there's a big challenge, which is transplant is thrown in the middle, which often will transplant as many different things. And that will often scramble the patient's journey along the way and make it difficult to generalize. But 2 factors probably can help at least overcome some of that. One would be, and this was brought up very nicely by David Miller, reproducibility. Reproducibility, my statistician colleagues remind me, is again something, if you have multiple data sources that are telling you roughly the same thing, that's something that's much more powerful. And of course, size is our friend. The more data points you can have that fit the characteristics, the better. That's a luxury we don't always find, and we can't always take advantage of in the rare disease space. But those factors usually allow us at least to put this into context. I agree with Martha. It's really tough to talk about generalities here because each of these data sets and each patient has a different journey. But again, going very back to, I think, our keynote speaker and some of the introductions, the way you deploy this data, the way that you want to try to overcome challenges is absolutely depending on who you're studying and what you're trying to accomplish. But again, having that prospective view of it, laying out and seeing whether, again, reproducibility within different data sets can be harnessed is a key factor. Again, some generalizable ideas, but all politics are local when it comes to these registries.
I love it, Greg. I always take those nuggets from your talk is you can't eliminate bias, right? So maybe on my note first, given that the challenge with the bias in the package, some of the disease registry, and also the real world challenge sometimes with disease registry, that we won't be perfect. Any thoughts from your side that can help the field advance, in overcoming this, bias and the challenges, so enabling a successful submission?
Sure, I could do that in 1 minute. No, Greg, I totally agree with you. I don't think our expectation is that bias is going to be eliminated. Absolutely, we understand. I don't think that you can guarantee that in most situations, even with clinical trials, actually. So the question then becomes how aware are we of these biases, and how are we going to address them, knowing that they exist, or quantify them in some way? So, I think for us, there definitely issues, that are important with respect to capturing as much information from patients in as consistent way as possible with as little missing data that is relevant to help us understand, how comparable are the patients, data that we're getting from a registry to the American public, for instance, and, are the data from the registry applicable to U.S. patients? Are they similar with respect to the underlying mechanism of the disease, the oncogenesis? Are they similar with respect to the types of treatment they received before getting, an intervention? After receiving the intervention, what, treatments happened afterwards? And could they be responsible for some of the effect we're seeing? And are those treatments comparable to what we would see, in US patients? So, as with anything, having as clear a picture about the patients themselves and their story, their clinical journey, helps us understand, if the data are applicable, and also if we're looking at this data, and comparing it to data from a clinical trial or other data, helps us gives us some sense as to whether we're comparing apples and apples to the extent that we possibly can. And having patient level data that's robust, that's capturing what we think is a generalizable patient population as possible, enables us to do analyses that will do propensity score matching to kind of help maybe account for any imbalances that we might see or any biases, or as we're sort of pressure testing the information. I think having that ability is really important for us to have in some cases. The principle of not fishing through a registry to get some information and then presenting it to us, rather actually having sort of this prospective development program where you have laid out the plan, this is the registry that we've chosen, this is why we think this registry is, or a combination of registries, are the best data sources, and this is how we're going to use this registry, in our, to show effectiveness, for example. Having all of that laid out and discussed ahead of time, also helps eliminate, some of the concerns that we have about getting at it. I think, not sure if it was Mike or Gregory said, get at what is truth. I mean, we all really want to know what is truth. Is this truly a drug effect or not? If this is a drug effect, how does this impact our patients? Is this making a benefit to our patients? So, having a strong understanding of the registry, having that transparency, I think, as Dave Miller was saying before, having all of that and having those discussions ahead of time help us, I think, best navigate, chart a course that I think we're as comfortable with as possible. I think when you have a big drug effect, that also helps overcome a lot of issues, right? So if we're talking about a marginal drug effect, then those issues become much bigger because then you have more uncertainty about, does this drug work or not? If on the other hand, you have a very large drug effect, then, sometimes some of those issues, melt away or become certainly less important.
That's really helpful. Thank you, Marcia. I think we also have some really interesting questions from the audience. So we do promise that to take on some of their questions. One question I think is really speaks to the heart and purpose of our job is the helping patients. Here to talk about actually a perspective of patient enrollment and recruitment. And the question is, how do you envision patient registry to help for recruitment enrollment of patients for future trials? I think, as the industry side, Greg can speak this better than me, that we're encountering finding those rare patients, either, from an effort perspective and also screening perspective. It's not that easy to enabling a sizable trial. Marcia, to your point, to then enabling we can conduct a very thorough, I would say, hypothesis and experiments to find that signal. So any thoughts, Mike, since you're seeing patients on a daily basis, maybe you can give us your take on that.
I think in this age of social media, Twitter, Facebook, et cetera, I don't think that's as big a problem as you think it is. I'm incredibly surprised at how many patients show up in my clinic and tell me all about me because I heard about me from some other person on their blog or whatever. They are incredibly plugged in because their life is on the line. So they know which company is doing what and whatever. And I believe that they understand that going to a place which enables them to not just help themselves, but help every other person with this rare disease down the line will be very important. So you have a disease in which people can travel. Of course, there's some that you can't. And especially in a rare tumor, they'll go seek a second opinion. And they will travel to sites where they feel they're getting the best care. And they will travel to sites that feel that they have access, not just to what you know, but what everyone in the field knows. So I truly believe in my heart and soul that yes, they're rare patients because the average is rare. But if you aggregate all these patients in this country and they are a force to reckon with, they'll come to the place that will make sense to them. So I think the challenge is to us to really construct the compelling trials to demonstrate to the patient. I'll give you an example. If you try to mount a placebo-controlled trial, my patients look me in the eye and shake their head and say, you're kidding me, right? Right? I want them on the drug, I mean, for instance. So they kind of come with their own biases and their own, right? Greg, this is one of them, right? So with their own sort of wants and needs. And so a challenge is up to us to construct a compelling trial for these patients, to construct a trial that will answer critical questions and that gives them the best shot at a response. So I think we can do this. And you know, to have us all come together to present that to really the ultimate audience, which is our patients, is not just a challenge, but it's a task. It's a must-do task.
Brilliant, Mike. Thank you so much. And maybe that flipped to Greg. I mean, knowing that you know, this sort of, I would say, real world challenge. I mean, maybe to Mike's point, and now it's finally eased quite a bit in terms of enrolling those patients in the study. Will be interesting to see. I mean, there's an audience question regarding, you know, how this to prepare this registration quality disease registry data sets. So because we know that quality also important. So any thoughts on your side?
Just from the standpoint of using registries,
prepare a high quality data package.
Yeah,
for registration purposes.
You know, the registries can be helpful when you're designing your study for registration. They can give you insights into who these patients are. And there are nitty-gritty factors like what prior therapies they had in reality and issues with con-meds, you know, really nitty-gritty things that we can help design our studies to be more inclusive rather than exclusive based on the registries. But the most important factor I think registries can get at is really there's a power, particularly when we have a molecular understanding of who these patients are. If there's a connectiveness to biobanks, to think about who are the right patients that we really want to be enrolling into our study. Where's the highest unmet need? And again, refining that population for whichever way it goes. Either they're being poorly served by what's out there, or potentially that they could be better served by this therapy. That would only have the opportunity to improve the effect size. And of course, nothing gets patients more excited to be on a study than seeing a good effect size of a drug. And so that's one, you know, I think one aspect that I feel quite passionate about, figuring out who the right people are to be on a study. We're not living in the age where histology is just defining the diseases anymore. And I think Mike and Martha have talked about this pretty nicely. And then finally, you know, getting patients onto studies from registries. I would say also that, you know, particularly the DNA sequencing companies that are out there, they're a form of registry in many ways. And they are trying to be used, whether it's as Mike points out, through a community, creating a community for patients to give them information, or whether you're pushing information out to them through those channels. It's a way to get rare molecular subtypes on the studies. With regard to the making registrational data more, you know, higher quality, I just had one thought when Martha was talking, I wanted to get back to, which was this idea of bias. How can you get rid of bias? Well, you know, we already said we can't get rid of bias. But if we have enough information and enough structured information, we can actually predict what missing data, if we're missing, how bad is it? And so, for example, in the ALL case that Martha was citing, in a related ALL case, you know, the data that we had allowed us to say something along the lines of, if 50% of the patients had an unmeasured variable, it would have to have an effect size bigger than any of the other known prognostic factors for it to change the result. And again, that gets back to Martha's point that big effect sizes are helpful as well. But I covered a lot of topics there, but wanted to at least loop back to that idea, which is we can quantify the impact of potentially.
That's so brilliant. And I know we have about 4 minutes left, and now means, you know, we're in the, you know, election season, that we have a poll on the side from the audience. Clearly, you guys done a fabulous job. Majority of the audience do see real-world evidence will become mainstream driver, more than 56%. I think the sample size is big enough to draw a conclusion there. And then the second thing is also they see this data can be used for synthetic control arm and the experimental arms for consideration, you know, in the submission. So we won't leave this meeting without a call to action. Maybe I'll go around the table and then again, start with Mike, but then Greg and finish with Martha, see what your thoughts for the field to move forward. And probably in a minute time frame. Mike, go ahead.
Just very quickly, I feel like it's a presidential debate, and I got 2 minutes. Just kidding. So you know, I'm filled with a sense of hope and optimism, and I want to pass that on to you because I see that things are moving. But in order for us to do that, again, we have to really come together as teams to develop compelling things that can help. I think other than us doing registries, the rest of the field has to come to us as well, including sort of new understandings of what sort of data is really needed and what sort of quality measures must be put in place. So we need a whole new set of standards. In addition, we need to engage with our mathematical colleagues. Are there statistical models that can help us understand bias? For instance, Martha said compare apple to apples. Well, maybe if we have registries that are spread so wide that it goes in the community and infiltrates every part of oncology, you can find the apple in a mass of oranges or something, right? So thank you. I thought you'd like that. I'm going to end there, but leave with a sense of optimism and hope because we are moving this chain. And if we ever have the next symposium on this topic, we'll come with data.
That's awesome. Thank you, Mike. Greg?
I would just add that, you know, as we think about this source of evidence, it's really important for the good of patients that of course, that we get as much as we can out of this. But the slippery slope is to take it too far and describe this data, you know, is not randomized information, and we have to be honest. And again, I think David Miller commented very closely, working with the experts in this field, whether that's statisticians or epidemiologists. There's a long history. I'm a physician, but that's not my expertise. We need to bring those people around the table to be able to fairly discuss what this data is and what it's not. Now, again, at one extreme, you have sort of the plural of anecdotes, which is not evidence. And at the other extreme, of course, we have information that can really help us not only design our studies, but interpret them on the back end. And of course, in scenarios where patients and time and dollars and physicians' time is just getting shorter and shorter, we have to make sure that we're asking the right questions in the right patients. And we're focusing on what matters. And that's where this data source can only help us.
Thank you, Greg. There's no better person than Martha to help us give a conclusion there a bit. Martha, you may be on mute.
I am. Sorry about that. But thank you for that. I get back to what Greg and, many people have said, before, is that, the purpose of registry is ultimately to improve the lives of patients. And so I think, one of the, things that, one of the calls to action needs to be, sort of continuing to carry the torch that PDS is carrying, and making sure that we have the opportunity to work together as a community to make sure that the registries are serving their intended purpose, and always keeping that in mind, bringing patients and advocacy groups to the table to kind of in partnership to make sure that these registries are resources that are used to the best of their ability to serve patients. So I think that to me is a really important action item. And I think a little bit hinging off of what Greg just alluded to, Dr. Pazder in the OC, one of his favorite things that he likes to say to us is, don't let perfect be the enemy of the good. And so, I think we have to keep in mind that, registries are not perfect. No matter what we do in the future, it's not likely, it's going to be messy to use to some degree data from registries and other real world evidence. I think we have to accept that and work together to kind of, to make sure that the information is used in the best way possible. And I think it can be to expedite drug development, both for patients with rare cancers and other patients. So I'll stop there.
Thank you, Martha. That's a wonderful conclusion. And I want to take the opportunity to thank my wonderful panelists. You guys make the moderator job fun again. So thank you so much. Okay, so stay tuned. I think the next one is the fireside chat with my esteemed colleague, Sean Cozen and Josh Blinker. So we'll have a very short transition. So stay tuned. Thank you all.